Welcome back to ASM Live, the talk show from the American Society for Microbiology meeting in Boston, Massachusetts. We're here to have conversations about microbes, stories that are important, interesting, and sometimes downright surprising. I'm Stanley Malloy, I'm your host, and today our guest is Otto Schwake. Otto is a student in microbiology at Arizona State University, and he collaborates very closely with people in engineering and the built environment. Otto's going to talk to us today about some very surprising places where we may find a human pathogen and ways that we may be exposed to pathogens that we never would have guessed some years ago. But before we go into this, maybe we can back up a little bit, and why don't you tell us what we mean by the built environment?
So basically, the built environment is any human structure, ranging from indoor air quality, surfaces, but also other engineered systems like water distribution systems, motor vehicles, for example. So there are a lot of different examples of what constitute the built environment. And an important aspect of it is the microbiology behind it. All environments are different. Built environments are no different in that they have a wide variety and diversity of microbes in them, with potential pathogens.
So this ties in, I think, very nicely to the discovery of Legionella as a human disease. Would you just quickly remind us about that story?
All right, sure. So I like to think of Legionella as a sort of good example of a pathogen of the built environment. These are gram-negative bacteria, in a lot of ways pretty standard, with some not so standard characteristics that make them sort of ideal built environment pathogens. They're what we call ubiquitous in fresh water. So pretty much anywhere where there's fresh water that doesn't have some sort of unusual characteristic that prevents them from growing, we're going to find them. I can almost guarantee you, for example, we'll look at enough tap water from this very building and we'll find some Legionella. Going back to when we first discovered it as a human pathogen, the original outbreak of this disease actually happened in Philadelphia, pretty famous at a Legionnaires, American Legion convention, hence the disease, Legionnaires disease, where the CDC eventually traced the outbreak to aerosols coming from cooling towers in the hotel where a convention was being held. Legionella likes water and it likes warm temperatures. So you combine those sort of aspects that can happen more often in engineered systems as opposed to natural water systems with aerosolization, we can lead to incidents of disease and outbreaks of this pathogen.
So we build an environment that pipes these organisms right into our lungs.
Pretty much.
And they thrive in that particular environment. That's a cool thing to do.
Right.
So where else is Legionella transmitted from?
So this is one of the interesting things about this disease. Not just the microbes, but actually looking at from an epidemiology standpoint, public health standpoint. They're, like I said before, they're everywhere. Anywhere where there's water, we're going to find Legionella. Unfortunately, there's a lot of dogma surrounding Legionella in regards to public health, where people sort of assume that a single species causes almost all the disease, and that only a small number of sources are responsible for transmitting Legionnaire's disease. The number one source, I'm sure a lot of us microbiologists are aware of, are cooling towers. Outside of cooling towers, spas, and fountains, people tend to sort of almost shy away from assuming that another source could be responsible, or at least responsible in any sort of major way for Legionella transmission. Unfortunately, this is shown to not be true, though. There's been a lot of documented cases from atypical sources. A good example is in the 90s, there was sort of a, I don't know if I want to call it a public health scare, involving dental water line units, where contaminated, the water that you get in your mouth when you go to the dentist, people were getting actually endocarditis from Legionella and other bacteria during dental surgery. Another good example I like to throw out is soil. They're actually soil-borne Legionella. And in other parts of the world, like New Zealand, for instance, where public health officials look in these sources, sometimes they find that these, what we consider atypical in America sources, tend to be very important sources of transmission. So in New Zealand, for instance, I believe 85% of Legionnaire's disease cases can be traced to potting soil and compost, whereas in America, it's, I think, 0.5%, something like that.
So another place I've heard of are the misters in the produce departments of supermarket. Very similar thing where you're pumping all of this water into people's lungs, right? So one of the key things that you mentioned just a little bit ago is the idea that they grow and thrive in this water unless the water's treated specially, right? So now you've discovered this very unusual source, a potential source of Legionnaires, but it's a often treated source, isn't it?
Not specifically treated for Legionella. It's unique and it's not just water, it's water with chemicals in it, some abiotic chemicals, but still, it's not specifically treated. So I don't know if it's kind of like the same thing as a treated water system, chlorinated water system, for example.
So I want to stop for a second and let anyone in the audience, if you have any questions, please join us in the conversation. Just come up to the microphone. And if you're listening on the internet, you can send us questions via Twitter at #ASM2014. So let's come back to this question. The place that you've recently found a substantial amount of Legionella is windshield wiper fluid.
Yes.
What have we found?
So I'd like to start by going back to a little bit of the justification for starting this study. It's actually been happening for a few years now, almost 10 years. Epidemiological studies have been showing pretty convincing evidence linking automobile use and Legionnaire's disease. And I've heard some anecdotal evidence from respiratory specialists, infectious disease specialists, respiratory diseases, sort of saying a lot similar things. But a few studies have been showing for a while now that professional drivers in particular have increased risk for pneumonia caused by Legionella. And looking into this, there's sort of three pathways that we see research groups looking into. One being puddles splashing up Legionella from the ground, the air conditioning systems in the cars themselves, and the third being washer fluid spray. And our group decided to follow the latter pathway to investigate. And I'm sorry, can you repeat the question one more time?
Well, that was a really important thing, and why you would even look at this place. But how common is Legionella in the washer fluid spray, now that you've looked at this?
All right, so we've actually done a field study in Arizona. Unique in a lot of ways. We don't get too much freezing weather in central Arizona. Our washer fluid does not have a lot of antifreeze in it. But we did a field study with elementary school buses looking at their washer fluid. We found some pretty high percentages of positivity, pretty high numbers. Just throwing a couple off the top of my head, I believe with culturing and DNA, combination of detection with both, we saw 83% of samples we collected had Legionella in them. 75% had Legionella pneumophila, which causes about 95% of Legionnaire's disease cases in America. What was really surprising to me was the concentrations. We saw ranges, of course, 0, but culturable numbers, so culturable, not just total cells, from 4 to 140,000 CFUs per milliliter. So one bus we looked at actually had almost 1 billion Legionella pneumophila inside of it. So pretty high numbers.
What's the typical dose that it takes to cause disease from an aerosol, for example?
That is a good question. And there's a lot of, I don't want to call it speculation, but a lot of work needs to be done to really nail down an infectious dose. With guinea pig studies, most of them, unfortunately, a little dated at this point. People are looking at an infectious dose of 1 to about 1,000 Legionella deposited in your alveoli to cause Legionnaire's disease. A typical concentration in air that we see for outbreaks ranges from about 10 to 200 colony-forming units per cubic meter of air.
So clearly there are enough organisms in the fluid to be a potential threat here, but most of the time when we think of our windshield wipers flipping back and forth on the window, we're not thinking about the water flying in our faces. Mm-hmm. And so how do you make that connection?
So this is actually a question that people ask me a lot is, oh, so if I just roll my windows up, I'm not going to get sick, right? But we all know cars aren't airtight, right? And different cars have different levels of airflow in them. For example, the standard car I like to talk about is the 1989 Mazda 121. Not exactly the pinnacle of automotive engineering, but it has an air change rate of about 50 volumes of the car per hour. So 50 times an hour, it is flowing in a lot of air into the car. That's driving fast with the vents on, with the windows up. So air is constantly being taken into cars, right? Depending on a lot of factors. And our idea is that even if your windows are rolled up, if you're spraying your cars and Legionella is being aerosolized, some percentage of it is going to be getting in the cabin and could potentially be winding up inside your lungs.
I thought you were going to tell us the story we've all had where you're sitting with your window down at the stop sign and the person next to you turns on their wiper fluid right in your window.
In your face, right?
It's a pretty common story as well. So you said earlier that I think something on the order of 70 to 80% of the school buses showed Legionella associated with the washer fluid. But how broad of a sampling was that?
So it was a relatively small study. 12 buses. We did two series of samplings, both spread out over about 3 months. One in the summer, temperatures reaching about 42 degrees Celsius in Phoenix, pretty hot. One going from winter to summer, where temperatures reached freezing. Phoenix is a pretty big range of temperatures, right? 12 buses we looked at. Each bus had about 2 samples every time we sampled. So a relatively small N value, but pretty convincing to statistics when we look at the percentages of positivity in the numbers we saw. Something else I'd like to toss out. 12 buses, obviously not everyone had Legionella every single time, but for every single bus on at least one sampling period, every bus had culturable Legionella at some point. So we can assume that it's not just an isolated, very rare phenomenon.
Are any of the bus drivers in this region, have any of them had pneumonia associated with Legionella?
You know, something I've been wanting to do for a while is a survey, a public health survey. Unfortunately, we haven't been able to get that sort of experiment off the ground, but it's definitely something I'd really like to look into. Pneumonia or just respiratory disease in general, compared to the general public, and see if they're higher or lower. Talking to a couple bus drivers I knew, none of them showed anything outside the ordinary, like colds and flus, no pneumonias though.
So when you aggregate all of these different sources of Legionella, from the produce department to the windshield wiper fluid to the air conditioning of buildings, it sounds like we're exposed to probably to a pretty substantial amount of Legionella over the course of our lives, every one of us, right?
Yes.
But we have limited outbreaks of Legionella pneumonia. Is that correct? Or are we missing a lot of infection that may just not hit the radar screen, or what do you think?
So the absolute numbers of outbreaks are pretty low. Legionella is responsible for more drinking waterborne disease outbreaks in America than everything else put together, than all viruses, bacteria, chemicals, and parasites combined. So, you know, relatively, the number of outbreaks are high for waterborne or respiratory disease. But it's pretty well assumed that the incidence of Legionella is very much underreported. This is a pretty common trend for pneumonias in general. Something like 80% of pneumonia cases never actually have a causative agent found. And combined with the fact that the vast majority of Legionellosis infections aren't actually pneumonia, they're upper respiratory tract infections known as Pontiac fever, we can assume that it's really more of a situation of underreporting. A good example where perhaps improved monitoring or public health practices could sort of address this issue is looking at New York State. So New York State is what, 10% of the population in America? About 60% of Legionnaire's disease cases are reported in this one state. And I can tell you it's not the water, or I can assume it's not the water. It's probably more the fact that they have public health policies in place to actually look for these. Reported cases in America, about 8,000. I've seen estimates as high as 150,000 expected cases per year in America. So I think it's a reporting issue.
Very, very interesting. We have a question from our online audience.
Yes, this question comes from the chat room. JKB wants to know, could we add something to the wiper fluid to kill the bacteria?
So that's a very good question. It's another one I commonly asked. In our studies, we saw that different washer fluids have different concentrations of chemicals. Antifreezing agent is what we believe to be the most antibacterial compound we're going to find in typical washer fluid. In America, we use methanol. Different countries use propylene glycol, ethylene glycol, stuff like that. Concentrations in America range from less than 1% to over 40%. And I've heard of extreme weather like Antarctica, Alaska, almost 90%, almost pure methanol. In lab studies, we saw that anything above about 10 or 20% kills Legionella pretty well. So you would think, oh, simple solution, just add antifreeze to all washer fluids. Unfortunately, it's almost a double-edged sword. Go to anyone who drives a very nice car and cares about their car and tell them to put high levels of antifreeze in their washer fluid and see their reaction. It actually is harmful to the car. It eats up the epoxy that seals windows and other things like that. In addition, some studies are coming out showing that methanol exposure from standard washer fluid can actually lead to levels, concentrations in the cabin of cars higher than the EPA's recommended dose. So it's almost like pick your poison. Do we want pneumonia or toxic levels of this volatile compound? I'm sure there's some balance that might be a solution, but at the moment, more research I think is needed.
What about sensitivity to things like chlorine or halogens in general? Is Legionella very sensitive to that type of treatment?
So Legionella, I happen to know Legionella's resistance to, be familiar with Legionella's resistance to typical water treatment technologies. They're, like I said before, in a lot of ways a standard bacteria. They do have a relatively rich lipid membrane that makes them slightly more resistant to radical oxygen species and other forms of stress. Chlorine, monochloramine, UV, they're slightly more resistant than, say, E. coli, which dies when it touches chlorine pretty fast. Unfortunately, Legionella have a lot of survival mechanisms. They love to associate within biofilms, and good luck killing bacteria with nearly any chemical when they're in a biofilm. Legionella are also endoparasites of eukaryotes, including amoeba. Legionella are known to be able to invade amoebal cells, and under certain stresses, amoeba can form cysts, which are basically immune to conventional chemical treatment. I think it takes something around Legionella inside an acanthamoeba amoeba to kill the Legionella with ultraviolet sterilization would actually start heating up the water. It would take so much energy. So under certain conditions, Legionella can be very resistant to chemical treatment.
Is it possible that within some of these sources, including windshield wiper fluid, the Legionella is within amoeba in that environment?
So we believe that is possible. We haven't tested for it directly, but we have sort of evidence supporting the idea that we know for a fact amoeba can be present in tap water, even treated tap water. And the fact that we're actually seeing levels of Legionella increase 3-4 orders from the tap water to the actual buses we sample, we can assume that there might be hosts, such as amoeba, making their way into the actual washer fluid reservoirs. A little caveat to that is that Legionella don't need hosts to grow. They're actually known necrotrophs. They can consume organic nutrients from dead and dying microbes as well. So it could be a combination of those two, but we definitely believe it's a possibility.
So you actually think it's growing inside of the windshield wiper fluid, not just persisting?
So that's what we believe. We haven't done specific tests to measure Legionella growth in pure washer fluid. We've demonstrated that a liter of plain tap water contains more than enough nutrients to support a healthy population of growing Legionella for over a year. and the fact, like I said before, highest concentrations we saw in the tap water that was used to mix up the washer fluid in our field study was about 100 CFUs per milliliter, and again, several orders in that higher we saw actually in the reservoirs. So we can assume that growth was occurring. Plenty of heat, which Legionella needs to grow, and also we consume plenty of incoming nutrients in the form of microbes or potential host in the tap water.
Again, I want to pause and invite anyone in the audience to please come up and ask questions. And if you're listening on the internet, send any questions by Twitter to hashtag ASM2014. So Otto, what next? So you've identified this unique environment, and there are a lot of these other environments we know about that are potential built environment sources of Legionella. What next?
So I mean, that's a very good question. And because there's such a paucity in information about Legionella, especially in regards to its presence in the environment, not just the built environment, all environments, its ecology, there's a lot of potential for research, different branches that take. Our lab in particular, we are taking sort of two approaches. We have a nice collaboration with a foreign group who are in the process of securing funding and organizing a nationwide public health investigation looking into different sources of Legionella transmission in their own country, including in washer fluid.
So which country is this?
Now, I don't want to say too much. Like, okay. I don't know how much they have planning. It's not America. It's a country where people care about Legionnaire's disease a lot more. Dropping some hints here. So our goal is to combine with more field study of our own, compare results to there, and see if it's a climate or a geography-based incidence of Legionella in this source. The other sort of aspect we want to take is using what information we have, using our field data, our lab data, and try to come up with some sort of estimate for risk of Legionnaire's disease from this source. And right now we're working on a quantitative microbial risk assessment model, taking into account all sorts of different factors like Legionella concentration in washer fluid, simple things like the percentage of aerosols that actually infiltrate into the cabin of a motor vehicle, inhalation rates, infectious dose rates, and we're getting some pretty wild ranges so far between needing about 1 minute in a car to several billion hours to become sick. Big ranges, a lot of distribution in these variables. With the best estimate right now, we believe around 10,000 hours in a car under normal, consistent conditions, average concentrations, what we saw in our field study, to contract Legionnaire's disease. And by the way, if that sounds like a big number, just take into account the fact that the average American drives 100 hours a year. Multiply that by the 250 million motorists, and it can actually be a significant source of morbidity.
If you live in Los Angeles, you're in deep trouble. We have another question from online.
Actually, we're going back to the chat room with this discussion of disinfectants. And JKB wonders, if you shocked it with chlorine, would that disappear over time?
So that's an interesting question. Again, I'll start off with the whole double-edged sword sort of thing. You don't want to be putting too many dangerous chemicals in this sort of solution that's going to be aerosolized and potentially reaching people, right? We don't want to be breathing in too much chlorine. Chlorination is a pretty standard method of treating Legionella, but unfortunately there are a lot of potential issues with it. For example, in hospitals where Legionella can sometimes cause serious problems when it invades and grows in the plumbing in these buildings, chlorination almost seems to be negatively correlated with Legionella, or positively correlated with Legionella risk. So when we chlorinate hospitals to remove Legionella, there's a sort of theory I've heard that not only does it lower competition, but it actually makes the Legionella go into survival mode, increasing their persistence in the environment, but also increasing their virulence. So while you might lower levels of Legionella, you actually might be increasing risk for Legionnaire's disease. And again, that sort of goes back to the whole idea of how do we treat it. We need to know more about these organisms to understand how we're going to treat them in all sorts of different environments.
So I think the question really reflected the idea that you give a shock of high levels and then dilute out, right? So that you wouldn't have the persistent high levels of chlorine with those potential negative effects. The answer is probably that it hasn't been done yet, right? So we have another question.
Yes, this one is completely unrelated, but it also comes from the chat room. And that is Carla B., who wants to know if there are any vaccines being developed against Legionella.
No vaccines for Legionella. Legionnaires disease infections are treatable. It's a bacteria. It's pneumonia, so obviously relatively high mortality rates. Without treatment, outbreaks anywhere from 5 to 30%. With treatment, survival rates are very high. So Legionella tend to not have very high levels or very common antibiotic resistance. They can be treated, but unfortunately no vaccines exist for Legionella.
Now, if you take a look at the general population and you just look for anti-sera to show people who have been infected but maybe not shown any symptoms of disease or reported disease, how widespread does it seem like the infection is just in a random population?
That would be a very good study for someone to conduct, and as far as I'm aware, it has not been performed. It comes to the issue of exposure to Legionella is very common, sometimes very high, and again, without incidence of disease. So the results might be pretty tricky to analyze, and maybe that's the reason why no one's ever been tempted to do so, to do studies such as this. I would imagine that it would be, again, difficult to analyze what the data actually mean, but that positivity for Legionella antibodies in typically healthy people might be relatively high, just because, again, constant exposure, frequent exposure.
Do we know if high antibody levels are protective against Legionella?
I'm not 100% positive. I do know that in the overwhelmingly vast majority of cases of Legionnaire's disease, antibodies are produced. The most common way clinically we test for Legionella is with a urine antigen test. Unfortunately, and this kind of goes back to a sort of dogma of Legionella from a public health standpoint, this test tests for a single serogroup of a single species of Legionella. So again, I think it's a factor in the possible underreporting of this disease. We're looking for one of the potential, I think, 70 species that can cause human disease.
But in the United States, that would hit about 95% of the cases. Is that the prediction?
Reported cases. But again, it might be a sort of self-fulfilling prophecy. If you only look for serogroup 1 Legionella pneumophila, you're only going to detect serogroup 1 Legionella pneumophila.
So you've had to focus on Legionella, but this is a great example of a pathogen that is also a normal environmental organism, right?
Exactly.
It's around us all the time, and really it's a matter of figuring out ways where we get exposed to it at high levels that are the cause of human disease. Do you know of any other cases of another environmental pathogen that may have a similar lifestyle and a similar type of exposure mechanism to humans?
Yeah, there's a few good examples. Just going back to your original statement, we live in an environment surrounded by microbes. They really dominate the planet. Most of them are harmless. A lot of them are harmful. And probably a lot we don't realize are actually causing human disease, and especially from sources we don't know about. To directly answer your question, a good example of an organism similar to Legionella in a lot of ways are Mycobacterium. So Mycobacterium avium is a waterborne pathogen. Similar to Legionella, it's often associated with being a opportunistic pathogen, even though it can infect healthy people under the right conditions. It's another intracellular pathogen. It has very high survival in water. It's a respiratory pathogen. It's another example of an organism that has existed in the environment for, assumingly long before humans were here, that's only recently been causing disease because of advancements in heating, cooling, and water distribution that's allowing it to get to us.
Well, I think that this has been a great discussion about an example of our understanding of microbiology in the environment and, in fact, new ways to detect microbes in the environment and to be able to maybe better understand disease and better predict sources of disease and change the way that we detect and respond prior to major outbreaks in the future. So it's a very exciting time, and we look forward to the rest of your studies. I want to thank our guest today, Otto Schwake from Arizona State University. I'm your host, Stanley Malloy. We invite you back to another session of ASM Live. Thank you very much.
Extending the legacy of a classic reference, EcoSal Plus joins the ASM family of periodicals this June. This growing collection of continually published reviews reveals our ever-expanding understanding of the biology of E. coli, Salmonella, and the other Enterobacteriaceae. From cell architecture and growth to pathogenesis of infections to life in communities, EcoSal Plus is the single source of knowledge about these organisms and their use as model microbes. A distinguished editorial board, led by Editor-in-Chief James Caper, ensures that content serves the needs of the research community. Free trials available for institutions. Take a look at ecosalplus.asmblog.org.